Hi, I'm Ma Luo. Today I'm going to introduce our paper recently published in JCI about a novel approach for developing an effective HIV vaccine. HIV is very diverse and mutates rapidly. However, the more challenging aspect for HIV vaccine development is that the virus targets an important part of the immune system, CD4 T cells, especially when they are activated. Because vaccines aim at generating immune memory that can be rapidly activated to destroy virus or viral infected cells, the activated immune system will include CD4 T cells. These activated CD4 T cells can help virus to establish infection. Can we generate anti-HIV immune response without increasing the targets of HIV-1, the activated CD4 T cells? We can learn from natural immunity to HIV-1 of a group of sex workers in Nairobi, Kenya. These women remain HIV uninfected despite high-risk exposure. Dr. Frank Plummer, the co-author of our JCI paper, established and maintained the Pongwani cohort. He and co-investigators identified this group of HIV-resistant women and studied their natural immunity to HIV-1. The research on the natural immunity to HIV-1 in this woman contributed to the development of the PCS vaccine. Sadly, Dr. Plummer passed away on February 4th this year. We dedicate this JCI paper to him. The study found that the spectra of their anti-HIV CD4 and CD8 T cell responses is narrow, and the magnitude is low. HIV class 1 genes determine the abilities of CD8 T cells to respond to infectious pathogens. These genes are very polymorphic, each with over 6,000 alleles in the world populations. Because of the variability, they have different abilities of recognizing pathogens. We found out women with AO1 is less likely to be infected, while women who have B0702 were rapidly infected after entering the cohort. To understand why, we compared abilities of these two HRR alleles in binding recognizing HIV GAG peptide. What we find is not as expected. The A1 allele associated with resistance to HIV infection can only bind one GAG peptide with high affinity. The B0702 allele associated with susceptibility to HIV infection can bind 30 GAG peptides with high affinity. Thus, for prevention from HIV infection, more might not be better. We noticed that the peptide recognized by O1 with high affinity is located right beside the HIV proteinase cleavage sites of P17 and P24, as seen in the right circle. Why this is special? To produce an infectious viral progeny, the viral polyproteins needs to be cleaved by HIV proteinase at 12 sites in a sequential and precise manner. If one site is not cleaved properly, the virus will not be infectious. So if we develop a vaccine that can generate immune response to the sequences surrounding those sites to force the virus to mutate, we could make the virus non-infectious. Since the sequences surrounding those sites are highly conserved, the vaccine can target all HIV subtypes. Because the vaccine-generated immune response is focused, it may avoid generating activated CD4 T cells, the easy target for HIV to establish infection in the host. We tested the vaccine idea with proof-concept preclinical studies in an HIV non-human primate model. The vaccine is consisted with 12 20 amino acid peptides. Each of them overlaps one of the 12 proteinase cleavage sites. The vaccine is delivered with recombinant VSU virus and a nano formulation. Our paper reports the recent studies. 16 female cynomolgus macaques were used. 8 were immunized with the PCS vaccine, and 8 were immunized with vector control. The monkeys were immunized 5 times. 6 months after the last immunization, we challenged the monkeys intravaginally with a very pathogenic HIV virus to mimic male to female HIV transmission. HIV MAC251 is a very pathogenic HIV and very difficult to neutralize. The low-dose repeated HIV MAC251 challenge is considered a very stringent way to determine the efficacy of a candidate HIV vaccine. The results showed that The PCS vaccine significantly protected the monkeys from a very pathogenic HIV infection. Because of the biological variations among the monkeys, another way of comparing vaccine efficacies is to examine how many HIV challenges to infect 50% of the monkeys. To determine how many HIV challenges to infect the vaccinated monkeys, we continued challenging the uninfected monkeys. The results showed that where it took only 2 challenges to infect 50% of the controls, it took 11 HIV challenges to infect 50% of PCS vaccine immunized monkeys. This is the best result seen for a candidate HIV vaccine tested with a non-human primate HIV MAC251 model. What other factors correlated with vaccine-induced protection? Our analysis showed that all 7 correlates of protection are T cells. We further examined the T cell responses 6 months after vaccination. T cell responses of monkeys without antigen stimulation represent the status of the monkeys before encountering HIV virus. It showed that the amount of viral target cells, activated CD4 T cells, are low in the vaccinated monkeys, and their amount of CD8 T cells associated with inflammation is also low. When the monkeys' white blood cells were stimulated with antigens, the PCS vaccine immunized monkeys have lower viral target cells and the lower CD8 T cells involved in inflammation. The PCS vaccinated monkeys have lower immune activation status. The PCS vaccine immunized monkeys have higher PCS peptide specific CD8 T cells that have better potential of killing viral infected cells, and higher T regulatory cells that can protect against abnormal immune responses. To understand how immunologic factors might act together to protect the vaccinated monkeys, we performed a loss analysis. The analysis identified 6 immunologic factors that are key to predict vaccine efficacy. All 6 predictors are PCS peptide-specific T cells. Among them, 4 are T regulatory cells. Based on the value of these T cells, the Lasso model can accurately predict vaccine efficacy. The Lasso model also showed that it is not a single immunologic factor in determining vaccine efficacy. It is a combination of multiple PCS peptide-specific T cell responses that act together to protect the vaccinated monkeys. Each of the 6 key predictors also correlates with many other immunologic factors, suggesting that these factors influence regulate many other immunologic factors, forming a protective microenvironment. To conclude, our study demonstrates for the first time that a vaccine targets only viral maturation but lacks full-length envelope and gag immunogens can prevent intravaginal infection in a stringent macaque HIV challenge model. The PCS-specific T cell responses correlated with vaccine efficacy. The PCS vaccine did not induce immune activation and inflammation known to be associated with increased susceptibility to HIV infection.